So leaky gut is sort of a loose term for deficits in the ability of the intestinal barrier to function properly. And so, one of the main functions of our gut is to allow certain molecules or nutrients in while keeping other more toxic agents out. And this is once again one of the most fundamental aspects of GI physiology. And what's been reported in human autism, in subsets of human autism that we reproduce in mice, is this syndrome that we refer to as leaky gut, where this discrimination of molecules that should be allowed to pass through the GI tract into the circulation is lost. And essentially many different metabolites or small molecules leak out of the gut into the circulation. And we furthered this research by identifying specific metabolites, specific small molecules that we believe are produced by gut bacteria that are getting into the circulation of the quote-unquote autistic animals. And then the final experiment that I think that convinced us that these metabolites weren't just sort of a bystander or a surrogate for leaky gut, but may be causal in the behavioral deficits, is that we injected one of these metabolites that was leaking out of the gut into the circulation. We injected one of these metabolites into healthy, normal mice and observed that that metabolite affected the behavior of what were, once again, otherwise healthy organisms, healthy animals. Yeah, so there are probably a variety of ways that gut bacteria can access the brain or brain function. And of course, we haven't studied all the different mechanisms by which this could occur. And so there's evidence that immune cells traffic from the gut to the brain. And so perhaps immune cells can pick up signals from gut bacteria, anti-inflammatory signals that suppress immune responses and can traffic to the brain. The nervous system is connected between the gut and the brain. So the enteric nervous system through the vagus nerve connects to the central nervous system, the brain and the spinal cord. And that might be a way that microbes can access the brain or brain function. And one area that we have, or one of the alternative mechanisms that we have tested, are that small molecules released by gut bacteria can travel through the circulation and potentially into the brain and affect brain function. So there's a variety of ways that these microbes can access the brain or the central nervous system and affect whether it's inflammation in the brain or behavior, perhaps even psychiatric and neurological and neurodegenerative disorders.
Do we know yet what a healthy gut bacteria ecosystem looks like?
Well, I think the field has done a fairly good job so far in terms of defining a healthy microbiome, the collection of organisms that live in the gut. But I think there's still a lot of work to be done. A lot of this work was spearheaded by the Human Microbiome Project, which surveyed many hundreds of people, not just in the gut, but in many different sites, and asked the question of what is a normal microbiome? What are the microbes that live in and on humans? And I think it gave us a pretty good profile or a catalog of the organisms that live in humans. But once again, it was in some ways limited because there's only so much that sequencing technology can tell us. And secondly, it's defined to a very small population, and that was the cohort that was used for this particular study. And I think it's important to remember that humans are quite diverse. They live in very, very different environments. They eat many different foods. And so sampling across geography, across age, across time may give us a very different picture in terms of what are the normal healthy microbes that inhabit humans. So I think there's still a lot of work to be done to define what is a healthy microbiome.